Good afternoon, everyone. Today I'd like to talk to you about the research that I'm involved with at the Fiona Relsey Cancer Research Institute in Ballarat, where we're trying to investigate the role of the immune system in fighting cancer. So the immune system is a really complex system consisting of many different kinds of white blood cells, which some of them are shown at the top of this slide here. You don't need to remember all the names of these different cells. What is important is that they all have very different functions, and they're all able to communicate with each other, and they work together to do their job. Of course, their main job is actually protecting us from pathogens such as viruses and bacteria. But there's also an increasing amount of evidence that suggests that these immune cells are also able to detect and fight cancer. So you can see down the bottom of the slide here some healthy, happy cells, and I want to show you now how the immune system responds when a good cell turns bad or undergoes malignant transformation to become a cancer cell. The most important cells in the immune system for fighting cancer are these ones here, the lymphocytes, natural killer cells and cytotoxic T cells. These cells are able to detect changes in the normal cell as it becomes a malignant cell, and they're able to kill the cancer cell. This is all well and good, and this happens very effectively in the early stages of cancer. The problem is that cancers can change. Cancers are able to mutate and change their appearance so that the immune system can't see them anymore, and they're no longer able to detect the malignant cells. It gets a little bit more complicated than this. There are also some other cells that make up the immune system which have a regulatory role. These cells are really important because they have the ability to control the function of the other immune cells. They can work in two different ways. We have some cells that act like cheerleaders and encourage these cells to do their job better and more effectively kill cancer cells. But there are also regulatory cells that can suppress the response of these effector cells. And so we have a balance going on between telling the immune system to respond and telling the immune system to stop responding. These are the cells that our lab is particularly interested in the context of cancer. We know that in some cancers, particularly solid cancers such as melanoma, cancers can secrete substances that actually promote the increase in these suppressor cells. So they're actually causing the brakes to be put on the immune response. And then the cancer can grow. The particular disease that I'm looking at is chronic lymphocytic leukemia. It's actually a cancer of a type of white blood cell. Why this cancer is particularly interesting in studying the immune system is that following diagnosis, patients can remain in a stable phase of the disease for varying amounts of time. They can progress immediately to an aggressive form of the disease, or they can stay in this stable phase for up to 20 years. And the big question here is what happens that causes people to go from the stable disease into the aggressive disease? And we think that the immune system is probably involved. So in the stable phase, the immune system's controlling the disease. Something changes that allows the cancer to grow. And we're actually seeing some really striking results when we analyse these cell populations in patients' blood. When we look for these stop type of cells, we find that they're increased in the blood of CLL patients compared to healthy controls. This is similar to what's seen in melanoma. Another interesting thing we've found, which is completely new, is that there's another type of regulatory cell that we think promotes immune responses, and this is decreased in CLL patients. So we think that the absence of these cells is contributing to the ability of the cancer cells to grow. So what does all of this mean? In terms of outcomes for patients, the changes that we've identified may be able to better predict CLL progression. One of the big problems at the moment is that we have no idea of telling when patients are about to go from stable to aggressive disease. And what we're seeing is that these cells are really strongly reduced. That might be a good predictor of progression. Importantly, it may also need new therapies that target regulatory cells so that we can shift the balance from a situation where we have uncontrolled cancer growth and the immune system is blocked and scratching its head to a situation where we have an active immune system that's able to control the cancer. There are two ways we can do this. We can block the effect of these stop cells, and there has been some success using these sorts of treatments in melanoma. Our findings suggest that these sorts of treatments might also be good for CLL. The other thing we can do, which would be a new approach, is to activate these cheerleader cells that will enhance the immune response to the cancer. And these are really important discoveries because they're bringing new hope to people with CLL that there might be a treatment in the future. Thank you.